Hello, everyone. I'm Rodney Varner, Chairman and CEO of Genprex. Thanks very much for joining the presentation this afternoon. Many of you, I know that many people watching have followed us for some time. Certainly others may not have. I've got a slide deck here that is on our webpage for those who would like to look into it in more depth. You're welcome to look at it there at genprex.com. Of course, at Genprex, we're reprogramming the course of cancer and diabetes through gene therapy. I know this audience has seen the forward-looking statement slide many times. I commend it to your reading. As you know, things may not always go as planned or as projected or forecasted, and it's important that you realize that. And I commend this slide to your reading on our deck on our web page. This is our stock data at a glance. Our market symbol is GNPX. Traded on NASDAQ. Don't know what the share price is right now, but the market cap has been around 150, 160 million, 39 million shares outstanding, and 10-day average trading volume of around 1 million a day. For those of you who may not have time to listen to the entire presentation, I hope you'll follow this slide with me. This is a summary of some of the major points of why we believe you should consider investing in Genprex. First of all, we have a gene therapy platform. We can use our lead gene therapy drug and delivery system with multiple other cancer-fighting genes. We can fight multiple kinds of cancer, and we can combine our drug with multiple other approved cancer drugs. We have, we believe, the first systemic gene therapy used for cancer in humans. And I mean actually used. We've actually treated over 50 patients in clinical trials with this gene therapy in cancer. We're addressing really large markets and large unmet needs. Our initial target is lung cancer with a drug market of in excess of $26 billion a year. And this point goes hand in hand with the next one, that we're combining our gene therapy with top-selling cancer drugs. We have 2 clinical trials planned to start the first half of this year. One combining our gene therapy, Recorsa, with AstraZeneca's Tagrisso. Tagrisso is AstraZeneca's largest selling drug with 2019 sales of over $3 billion. One of the clinical trials will combine with Merck's Keytruda. Keytruda is Merck's largest selling drug with 2019 sales of around $11 billion. So these are very large selling drugs that we're combining our drug with. And note that we're not competing with them. We're not trying to take market share from them. We're trying to be added and combined with them and to increase their markets as well as ours. We have an FDA fast track designation for use of our gene therapy, Recorsa, with Tagrisso. That's AstraZeneca's drug that I just mentioned. We have demonstrated clinical success. We've treated over 50 patients over multiple clinical trials. In those clinical trials, we've shown a favorable safety profile and strong indications of efficacy. As I mentioned, we're initiating these 2 clinical trials here very soon. We have world-class academic partners in MD Anderson Cancer Center for our oncology program and the University of Pittsburgh for our diabetes program. We have an expanding pipeline as evidenced by our diabetes program, which we brought into the company last year. Early last year, we in-licensed that technology from the University of Pittsburgh. And we have a strong balance sheet. we raised around $40 million in 2020, all at the market and with no warrants over 3 rounds. The most recent round in January of this year, December of 2020, was $12 million from a single healthcare dedicated investor that had followed us for some time. This was a single institutional investor who is very sophisticated in the healthcare markets. Along with the balance sheet, we've got other strong indications that our message is being heard and we're gaining traction in the markets. At the beginning of 2020, we had fewer than 5,000 shareholders, and I just got a count very recently. We now have over 20,000 shareholders. At the beginning of 2020, we had only a handful of institutional investors, and now according to the forms that they file with the SEC, we have a large number, probably dozens. So we're seeing real momentum and traction in the markets and recognition that we have a strong program, that we're on a course to success with the FDA Fast Track designation, and the other work that we're doing with our 2 clinical trials. So a bit more on the Fast Track designation. I mentioned that it's with Tagrisso, that is to combine our drug with Tagrisso. The way this would work is that as patients take Tagrisso and become resistant to it, and by the way, nearly all lung cancer patients do become resistant to Tagrisso. It's nearly 100%. According to AstraZeneca's data, the median time to progression on Tagrisso is between 14 and 18 months. So our Fast Track population is to treat those patients who take Tagrisso, they become resistant to it, they're going to need another drug at that point, and we would come in, combine our gene therapy Recorsa with Tagrisso, and continue to treat those patients. That's what the FDA has approved as our Fast Track population. I've got some additional information on the design of that trial later in the slide deck for those who are interested in really getting into the details. Now, I have people ask me, what is a Fast Track designation? What does it mean? Well, the FDA awards the Fast Track designation after an application is made that presents all of the company's data, both preclinical and clinical, and the FDA must find that the drug has a potential to be an improvement over existing therapies in an area where there's an unmet need in a serious or life-threatening disease. Well, we're gratified that the FDA made that finding for our drug, and that that population is late-stage lung cancer, which is a very large market with huge unmet needs. The Fast Track designation helps facilitate drug development. It provides pathways for potential expedited regulatory review and potential accelerated approval, additional attention from the FDA, and basically a recognition that the FDA believes this is an important drug that needs to be gotten to these patients in need as soon as possible. This slide illustrates our pipeline. We have the 2 clinical trials that I've mentioned, which we branded. The Tagrisso trial is branded as a Claim 1. The Keytruda trial is branded as a Claim 2. As you can see, they're both ready to start the phase 1/2 clinical trials. We previously had a phase 1/2 trial combining our gene therapy with Tarceva, which is an earlier generation EGFR inhibitor. The phase 1/2 data from that trial was instrumental in getting our fast track designation for the combination with Tagrisso, which is a later generation EGFR inhibitor. I think the FDA recognized that Tagrisso, the later generation drug, had largely taken over the market in the United States for this type of drug, EGFR inhibitors in lung cancer, and agreed with us that that would be a better combination for our drug. So that's the way we're going. Now I want to mention our diabetes program as well. This is early stage, it's preclinical. It's ongoing at the University of Pittsburgh, and I'll have a bit more information about it later in the deck. Here are additional details on our trial combining our gene therapy with Tagrisso, which we have branded as the ACCLAIM-1 trial. And our drug, by the way, it has an FDA-approved brand name of Recorsa. We have previously in our materials often referred to this drug as Oncoprex, but now our approved FDA name is Recorsa. For that same gene therapy, we now refer to our delivery platform as Oncoprex. So this ACCLAIM-1 trial is going to target patients with stage 4 EGFR mutant non-small cell lung cancer whose disease progressed after Tagrisso. And again, nearly all of them do progress. We're planning to have 10 U.S. sites. We are targeting to treat 100 patients, 9 to 18 in the phase 1 portion, 82 in the phase 2 portion, and we're planning an interim analysis at 53 events, which is disease progression or death. And again, we're initiating this study the first half of this year, and we're on track to do that. I'll leave the additional details of this study to your further reading on the deck, or if you want to get into it in the Q&A, but it is here in the deck. So just to be clear, we have two ongoing gene therapy programs. The one in oncology, which is I've spent most of my time on so far, is a gene therapy that was invented and has been developed at MD Anderson Cancer Center. There is both clinical and preclinical data that it can be combined with EGFR inhibitors, and we've shown a favorable safety profile and indications of efficacy in lung cancer. There's also a great deal of preclinical data indicating that this gene therapy for cancer may be effective in a wide range of cancers, may be combined with a number of other cancer-fighting drugs, and so, you know, we believe that it's an important platform in cancer. That program uses a non-viral delivery system. It's a lipid nanoparticle delivery system. Now, completely separately, we have a gene therapy for diabetes invented at the University of Pittsburgh that uses a viral delivery system, and it targets cells in the pancreas to convert non-insulin producing cells into insulin producing cells, with the goal of eliminating the need for insulin treatment for diabetics. Diabetes is a huge drug indication. It affects about 34 million people in the United States and very large numbers worldwide. So back to the cancer program. Non-small cell lung cancer, which is our initial target, well, lung cancer itself is the most common, lung cancer is the second most common form of cancer, and it kills more people than any other form of cancer. Non-small cell lung cancer represents about 84% of lung cancers, There are roughly 2 million worldwide cases a year, 228,000 cases in the United States. And if you look at the market size, as I mentioned before, Tagrisso, which we're combining with, $3 billion plus, Keytruda, which we're combining with, $11 billion plus, total market projected to reach about 26 billion by 2023. So vast markets. Back to our platform. This slide illustrates our platform. We can use our Oncoprex nanoparticle delivery system to deliver a number of different genes. There's a wide range of genes that can be delivered with these nanoparticles. We can deliver these genes to fight multiple types of cancer, and we can combine our gene therapy with multiple other cancer drugs. This is an illustration of the nanoparticle system. These are cholesterol molecules, but just picture fatty molecules. They have a positive electrical charge that is opposite that of cancer cells. They're injected systemically by IV, and they circulate through the body and attach to cancer cells wherever they're found. So this has the benefit of potentially targeting cancer cells that can't be reached by surgery or radiation, or may even not be known. This has been shown to be well tolerated in humans. As I mentioned, we've treated over 50 patients in our clinical studies with it. I'm going to skip this one. It's a little more detailed than I want to get into, but for those scientists out there, this may be interesting for you. This one as well. Our gene therapy, Recorsa, also modulates the immune system. This is important because it really provides a mechanism of action that supports both our EGFR program, that is combinations with EGFR drugs, and immunotherapy drugs. We conducted a phase 1 clinical trial using our gene therapy as a monotherapy, and using as a monotherapy, we showed that it halted cancer growth in 5 out of 23 patients, and we had at least stable disease in those 5 responses, that is reductions in tumor size in 3. And this established the maximum tolerated dose for the drug and showed that the drug was well tolerated in patients. We previously did a phase 1/2 trial combining our drug with Tarceva. I'm not going to get into all the data in this slide. Again, I would commend it to those who really want to get into the detail out there. But in this study, we combined our drug Recorsa with Tarceva, which is a first-generation EGFR inhibitor, sort of a predecessor to Tagrisso that we're using now. And in this study, we showed that our drug provided benefit in a number of patient populations that you wouldn't have expected benefit. All of these patients had had extensive prior therapies and had already exhausted all other therapies available to them before they went on to our trial. And even so, we got a complete response in one patient. We had significant tumor reductions in several other patients. And we saw patients benefit on EGFR inhibitors who had already failed them, which is not expected. So in the phase 2 portion of this study, 7 out of 9 patients received a benefit, that is stable disease or better. This study, along with our prior preclinical research, laid the basis for our FDA fast track designation. After receiving this data, we put together an application for fast track showing this and all our preclinical data showing all of the benefit in the various situations that I've described to you, and the FDA agreed with us and granted fast track designation for our use of our gene therapy with another EGFR inhibitor, Tagrisso. MD Anderson has done several studies of the combination of our drug, Recorsa, with immunotherapies. This one shows a study that was done in mice. These are syngeneic mice, or in my non-scientific mind, or regular mice. This study showed that combining our gene therapy with immunotherapies led to a better result. The mice lived longer. And it really applied to all of the immunotherapies that are out there in the market for lung cancer now, the various different types of them. So the combination of our drug Recorsa with these immunotherapies led to a better result than either drug alone. You can see this visually with the slides of mouse lungs on the right side. You can see the non-treated lungs and the treated lungs, and the treated ones simply look a lot clearer. Following the study that I just described, MD Anderson did another study with humanized mice. These are mice who have had human immune cells implanted in them where they grow. And it allows the scientists to challenge human immune cells with the disease, and then treat it and see what the results are. And in humanized mice, they essentially duplicated the results. They showed again that Recorsa, our gene therapy, is synergistic with immunotherapies such as Keytruda, and importantly, it also proved to be synergistic with the combination of Keytruda plus chemotherapy. So a three-way combination of Recorsa, Keytruda, and chemotherapy is more effective than Keytruda and chemotherapy alone. That's important because many patients who receive an immunotherapy like Keytruda also receive chemotherapy in combination. So if we're able to improve outcomes both with Keytruda alone and with the three-way combination with Keytruda and chemotherapy, we would cover most patients who are treated with Keytruda, this very large-selling drug. Now moving to the diabetes program. As I mentioned, type 1 and type 2 diabetes is an extremely large market. Many people in the US and around the world are affected by those diseases. This gene therapy, invented by Dr. George Geddes at the University of Pittsburgh, may potentially replace the need for insulin treatments in many of these patients. According to the researchers at the University of Pittsburgh, it could potentially even hold a cure for many of those patients. This uses a novel infusion process to basically inject adenovirus containing two genes into the pancreas, where they transform alpha cells that don't produce insulin into what they call beta-like cells that do produce insulin. And interestingly, these beta-like cells are not actually beta cells, and they're not attacked by the immune system. It's believed that the fact that they're not actually beta cells and not identical to natural beta cells allows basically the immune system to not recognize them and therefore not attack them, at least certainly not as quickly or aggressively as they would normal beta cells. In preclinical studies, these are mouse studies, this gene therapy has restored normal blood glucose levels for an extended period of time in the mice. Right now, research is going on with this gene therapy in apes at the University of Pittsburgh, and the researchers there recently received a $2.6 million NIH grant to support this research. We view that grant as very important in that it validates the research. I'm sure that most of you know that those grants are heavily peer-reviewed and very competitive. So not only did we get third-party validation, the university received additional funding to do additional work and potentially work that we might have needed to pay for. So this was a very positive event all around. I want to address some of our milestones. This deck is a little bit out of date. Manufacturing is very important. I'm sure most of you recognize that in gene therapy, manufacturing is often a huge challenge. Many gene therapies have died on the rocks of manufacturing, and in our case, our drug had been manufactured in several steps. In one, the DNA plasmid is manufactured, and then that is transferred to another manufacturer where it's formulated with the DOTAP cholesterol molecules and put into the final packaging for treatment. The plasmid has for many years been manufactured at outside contract manufacturers without a lot of difficulty. They've been able to manufacture consistently and at scale. Up until fairly recently, the second step of formulating the plasmid with the cholesterol molecules was done at small scale at the University of Texas MD Anderson Cancer Center. They have a GMP facility for research purposes, and the manufacturing was done in small volumes and for use on patients to be treated immediately after the drug was made for them. So we had to transfer the second step from MD Anderson to contract manufacturers and scale it up so that the drug could be manufactured at larger scale and shipped and stored for use in patients. We've now accomplished that, and we've recently announced that we completed the transfer from MD Anderson and the scale up, and that our clinical manufacturing run has passed all release testing. So this is a really substantial accomplishment that I'm very proud of for the company. We're continuing our preclinical research both in cancer and diabetes and potentially other indications. We're preparing for our 2 clinical trials that I've described to you. And as you can see, we did obtain last year, 2020, our brand name for Recorsa, our brand names for our clinical trials, and our non-proprietary name for Recorsa, all through the FDA. So we think it was a very successful year. People often ask me about comparables. We don't like to throw out other companies as comparables. We're not aware of anyone else doing any technology like we are. We are targeting lung cancer and diabetes, 2 huge markets, and we put up these companies, which you can see in our slide deck, as some of them in lung cancer and other solid tumors, and you can see their valuations, and we believe that based on everything I've told you, that we're seriously undervalued in this market and with the programs that we're aggressively pursuing this year. Well, I think that will conclude my presentation. Are there any questions at this time?
Hi, Rodney. That was a great overview of the company. We did receive some questions, and I also would like to emphasize some of the developments or would ask some of my questions. So first of all, congrats on the developments, the achievements you accomplished, manufacturing, branding, and also you're going after 2 really large indications that will be initiated in the first half of 2021. So my first question will be, considering you are going after Keytruda and Tagrisso, They both are, the mechanism of actions are very different. So PD-1 checkpoint inhibitor versus EGFR, do you have a stronger scientific rationale that you think there will be, one of the combinations will work better than the other one? That's my first question.
Well, first, Ahu, remember that I'm not a scientist, and I repeat what scientists tell me. And from my conversations with our leaders at MD Anderson and elsewhere, there's a very strong rationale for both of these combinations, and some of the rationale applies to both, the immunomodulatory effects, for example. So I don't detect from them a belief that one is necessarily stronger than the other. They believe that they're both very strong candidates.
That makes sense, Rodney. So my other question, follow-up question will be, when should we expect data readout from this initiation of clinical trials? Do you think we would have some data within 2021, or is it going to be a later at time type of data readout?
Well, we expect to have data from the phase 1 portions of the trials certainly in 2021. We haven't announced the time for the interim readout for the phase 2 portions of those trials. The Acclaim 1 trial, the one with Tagrisso, the readout is scheduled to be after 53 events. That's progression or death, which might be in 2021, could well spill over into 2022. It's a similar, there's a similar design for the Keytruda trial. We haven't completely finalized that design yet, so we haven't put it in the deck. But again, we're not giving a target for when that readout will be, but it'll be a similar number of patients.
And you also mentioned about the triple combination, chemo, PD-1, PD-L1 agent combination. Is that something you would like to pursue? Is it a strategy you would like to perhaps discover later in time?
No, it is a strategy that we're pursuing.
That's great. And I also don't want diabetes program to go missing, so I would like just for you to maybe give a bit of a roadmap. When you would commence the trial, when do we expect any data from that program?
Well, Ahu, I'm sorry, but it's really too early for us to predict when we'll commence that trial. As I mentioned, research is going on right now at the University of Pittsburgh in primates, and the researchers there are enthusiastic about it, and we're quite optimistic about the program, but it's really just too early to predict when we would start that trial.
That makes sense, Rodney. I also would like to emphasize the value creation that you mentioned. You increase your institutional client base, but I also would like audience to know that you actually, your market cap increased over 1,000% in 2020 timeline. So congrats on that. And my follow-up question on that would be, do you think you'll continue this growth for the investors?
Well, thank you for pointing that out. We're obviously very happy about that. I'm a lawyer by background, and I'm not going to predict that our market cap is going to continue to grow at that rate. We're very optimistic. We think we're very undervalued. We think that this is a great point to get into our stock. So that I'll have to leave it at that.
As a covering analyst, I would actually confirm that comment. I also believe it's a good point for investors to come in prior to commencement of clinical trial and data readouts. One question we received, and I think it's a very good point to make. There are two parts of the question. One of them is, how do you feel about how the market is valuing the company? And the second part of the question is, what would you like us to leave with from this presentation?
Well, as I said, we think we're seriously undervalued. We think that with 2 programs in lung cancer, probably the largest indication in cancer, and a preclinical program in diabetes, all with world-class researchers, that we should have a much greater valuation. We have a very strong management team. In 2020, we added greatly to our management team. We brought in 2 senior biotech leaders with over 30 years experience, a clinical trial manager with over 20 years experience. So we have very strong management to pursue these clinical trials. We have a strong balance sheet, having raised roughly $40 million in 2020. And the FDA Fast Track designation, a rapidly growing investor base, both retail and institutional. So, you know, we just think we're seriously undervalued. I hope that others will take a look at this and agree.
Thank you so much for your time, Rodney, and we appreciate your participation in our conference. Thank you.
Thank you, Ahu. I appreciate the opportunity.